Hi, you guys. This is the Unit 3, Segment 3. This is the second video. I went a little over 15 minutes, but I heart you people, and I know that you'll be fine. This will just be a few minutes. Anyways, I had to cut this short a little bit, but I ended with this slide. So we're looking at hypotonic solution on the left here. Hypo, look at that circle. It's swelling up the cell. Why does it swell up the cell? Because the concentration of water is higher outside the cell than inside the cell, so water will move in. And it's called lyse. The cell can burst, explode. And this is an animal cell. This would be a plant cell. You can see how it's not so boxy. It actually bulges out. And then the middle scenario, you have equal amounts of water going out, equal amounts of water going in. This is an isotonic solution. There's no net water movement. The plant cell will maintain its boxy shape. All is good in the world. Hypertonic, with an ER, not an O. Hypertonic is when the cell will shrink. Why does it shrink? Because water's leaving. So it goes from a higher concentration to a lower concentration of water. So water exits the cell shrinks as a result. In a plant cell, the cell wall will not shrink. The cell wall maintains its integrity, but the cell membrane pulls away from the cell wall. So you can kind of see there's pockets in there. And that's what happens when you put a plant in salt water, for example. The water will leave the cell because the salt water is making the water concentration lower outside of it, and it's higher inside, so it will leave. Okay, so far we have covered diffusion, just plain old diffusion, where a material will go from high concentration to low concentration. Then we talked about the diffusion of just water. And it has a special name, and is called, you know it, osmosis. And then now we're at facilitated diffusion. What happens if the molecules are too big or can't fit through the cell membrane, the phospholipid bilayer? What happens if the particles can't fit through that skinny little space? They need a little bit of help. So this material out here, this solute material, is higher concentrated on this side of the cell membrane and less on this side of the cell membrane. So this stuff wants to go through to follow its concentration gradient from high to low, but it can't get through. So it needs these proteins, okay, these channel proteins. They allow it to go through and move it through. There's no energy that's needed for this, guys. There is one that does require energy, but this is not that one. So facilitated diffusion requires no energy. It just allows larger molecules to fit through following its concentration gradient from high to low. The last one is active transport. This is active. Think active requires energy. Okay? This uses extra energy. It's active. These are going to go against the concentration gradient. Instead of high to low, these guys are going to go low to high, and there's reasons why your body wants to do this. So if your cell receives a signal that it needs to start pumping out salt, for example, then your protein channels will go ahead and start pumping that out. So it needs a pump. It requires ATP, which is the energy molecule. So this right here, this is a picture showing movement of these square solute materials against the gradient. It's higher over here and lower over here, but here is this guy moving this material in this direction. Just as a recap review, diffusion is particles moving through that can fit through from a high concentration to a low concentration. Facilitated diffusion is, it needs a little help to get through. It needs channel proteins to help it, but no energy is required. So these combined are an example of passive transport. Passive doesn't require energy. Active does. Another type of active transport, there's two. Active transport requires energy, so the cell is using ATP, needs lots of mitochondria to provide that. But endocytosis is actually when the cell engulfs or takes in materials, and those materials can be solids or they can be liquids. So endocytosis is when there's material going into the cell. I would actually draw this in the notes if I were you. two types of endocytosis, phagocytosis and pinocytosis. So phago is food. Phago, food, this is the cell taking in solids. So you've got cell membrane wrapping around this material and bringing it into the cell. This requires energy. It's a form of endocytosis, which is also a form of active transport. Pino, P-I-N-O, pinocytosis is taking in liquids. So you can see that the cell membrane kind of loops around, forms a little circle, pulls in the liquid, and then off it goes in this little vesicle and gets delivered to wherever it needs to go. So pinocytosis is liquid, phago is food or solid, okay? Make sure you know that for the test. All right, so endo is bringing in material. Exocytosis, exo, exit, is how things leave. So here's a vesicle that's floating around inside the cell, and it attaches to the cell membrane, it breaks apart, and then releases what was inside of it. So exocytosis is materials exiting the cell. And that is it.